So we're going to talk about B-cell biology and T follicular helper cells. Our goal is to understand how antigenic selection molds B-cell pools, and this will overlap a little bit with one of the earlier talks, but we'll look at it from a bit of a different perspective. We're going to talk about B-cells in the periphery and how we mold better and better and better immune responses, and why B-cell antigen receptors are really so much better than T-cell receptors. Boy, those T-cells are pretty dumb. And I like B-cells the best. And we'll talk about how we select these responses and about how mechanisms can subvert germinal center responses and how this may all relate to breaches in immunologic tolerance and autoimmune disease. I love this figure. And so this is very illustrative, I have to say. My children are a little bit older, but just a few years ago I was going over the chapter with my son that had to do with the earliest forms of animal life, and I was refamiliarized with the protists, and I have to say I found it quite disconcerting because I thought a protozoan was good. It was just so primitive that there wasn't much else to say, but they've turned them into animal-like protozoans, plant-like protozoans. It's so annoying. It's just... But so this has to do with evolution of the species, and I embrace all of my early precursors here. And so there are plants, fungi, and animals. And the reason I love to show this is just to say that, you know, with all of the species on the planet, very few really have or need an adaptive immune system. It's really a latecomer to the system. It's just a couple of 100 million years old. Bless you, bless you. So most animals and plants and fungi, they don't even need the adaptive immune system. In fact, you can be a very, very large multicellular organism. You can live for 100 years, and that's without a single lymphocyte. And the point is that with the burden or the benefits of the adaptive immune system and the ability to respond to new antigens, even ones that the species has never seen before, you also have a burden where you may develop autoimmune disease. And this is true for many mammalian species, as we will discuss. And often people will take me aside and they'll tell me about a new species that I should know also gets lupus. And so cats and dogs, I know somebody who actually submitted a proposal recently that had to do with studying the microbiome of pets and their owners that both have lupus. And they were going to find the key to it all, but that hasn't been published yet, so I don't know. So the idea is that there's a really basic principle which has to do with understanding how the adaptive immune system works, because it's quite a puzzle, as you'll see, having a lymphocyte that has a randomly generated antigen receptor that it's generated, and it doesn't really know what it's going to be selected against ultimately. And conceptually, it's quite intriguing, and it does relate to Paul Ehrlich's concept of the horror autotoxicus. And I have to ask William to give me a picture of that very attractive picture of Paul Ehrlich in his prime. I find that very impressive. So he thought that the adaptive immune system was a threat to the host, and how could it be that this would exist if you could have a reactivity to anything? And so he developed this hypothesis, and I have to say that these people were brilliant, these early immunologists. They were more like philosophers. I guess they were probably, in a previous age, would have been rabbis, that they could just argue forever and they wouldn't need any data at all. But Brescher and Cohen, and Mel Cohen actually, I hope he's in good health. He was about 92 last I saw him at the Salk Institute. They developed the two-signal hypothesis, and it's really very, very simple, even though it's 45 years old. Basically, you develop a lymphocyte from a precursor. It develops its antigen receptor randomly, and will it actually survive and get into the periphery? Well, autoreactivity is really a threat to the host. And the concept is the first signal would be the antigen encounter. And that will cause, theoretically, some level of stimulation. But actually, it will die unless it gets a second signal, because you assume that a lot of newly generated lymphocytes will run around the bone marrow and counter things that it might be sticky to, and that would actually be the flashpoint for an autoimmune disease unless there was a way to edit out autoreactivity. And basically, the lymphocyte, for it to survive, needs this second signal here. So first is antigen receptor encounter. If there is no second signal, that cell actually dies from activation-induced cell death. So the system weeds out in the central compartment, the bone marrow, all autoreactive clones. And if there is a second signal, then it can actually survive and proliferate. So that's an essential concept in immunology. There's a signal 3, which will actually cause the lymphocyte to be biased in one direction or another. This is true for T cells as well as B cells. For a T cell, this may actually bias to Th1, Th2, Th17, and it actually explains how from humble beginnings, a great diversity of lymphocytes with predominantly helpful binding specificities result. It is an untold secret that, in fact, every lymphocyte to even get past this level has to encounter something in the bone marrow that stimulates a little bit. It needs a touch or a tickling of autoreactivity. And so the thymus and T cells, they stimulate a little bit, but if it doesn't get a second signal, they die, and so in the bone marrow with B cells as well. So I think of everything primarily as a primate and secondarily from a broader view. And I don't know what my wife thinks I think about things as, but I'm not sure. But from the earliest beginnings of the adaptive immune system, it's only about at the hagfish stage that you actually develop antigen receptors that are the precursors of lymphocytes that uses combinatorial diversity to put together a limited range of building blocks to make more complex receptors. And you have to think about somewhere around the 1920s or 30s or 40s, when they began to appreciate that there were different immunoglobulins and antigen receptors that B lymphocytes made. The first hypothesis was that for every antigen receptor there might be another gene, and that would all be germline encoded. And so actually that seems like a good idea, except we know we can make lots of antibodies and lots of antigen receptors, but actually does anybody know how many genes we have? Because they didn't know at the time. You can yell this all out, and I mean, you guys are all mavens of the Human Genome Initiative, I hope. So it's around 30,000. It might be 23, but somewhere in between then. So we really only have a few building blocks. We can put them together different ways. For antibody genes, we actually have a couple of hundred. And from a couple of hundred, we can splice together numbers that are bigger than Carl Sagan numbers. They're bigger than billions and billions. They're just trillions. In fact, they're just crazy numbers. So that actually first arises as a hagfish or a shark. You are what you eat. I like sharks. But it turns out that only late in evolution, with the emergence of mammals, did our adaptive immune system and combinatorial diversity system actually become refined so that we develop what we're going to discuss towards the end of the program, germinal centers. So the germinal centers are these fantastic architectural constructions in our secondary lymphoid tissue that can be thought of as a maturation chamber or an arena of competition. And Garnet Kelso, a very good friend of mine, always described germinal centers as you would actually have one cell that would be, one clone that would be stimulated. It would make two cells. They would have a little bit of difference, and they would go one-on-one and have tremendous competition. So it's as though it's a gladiatorial bout for B lymphocytes. So that's just foreshadowing. You'll see what will happen next. This is the combinatorial antibody system. They actually should have put the heavy chain at the left because cells in earliest development, pre-B cells first rearrange the J mini gene segment to the D, and you have a number of these gene segments that can be accessed, and you just shuffle them and randomly decide which ones you're going to fuse together. And then subsequently, you'll pick one of the many variable region gene segments, and then you'll shuffle them together. And ultimately, you'll have a receptor, and if it is not autoreactive, it'll actually go on to then have the light chain loci. And if kappa loci doesn't recombine effectively, if you have something that's out of frame and for other reasons autoreactive, it will actually go to lambda. So there's a sequence, but it's a random sequence. And ultimately, there's goodness of fit, and rearrangements are proofread to see whether they're helpful to you. So from a limited number of germline segments, you could make lots and lots of genes. I have to say that was a linear depiction. What the heck does this have to do with an antibody that folds on the surface of the cell and becomes a receptor? Well, it turns out that there's a stereotypic fold. So I think about things in 3 dimensions sometimes, and if the gene itself looks like this as it's rearranged, and this is just supposed to be for a heavy chain, they actually found that all antibodies and all T-cell receptors fold into a beta-pleated sheet, which is something that's silk-like as well, and these just folds go back and forth, and they have loops in between them so that they can back up and go in the other direction. And it turns out the actual antigen contact sites are where the loops are. They're more flexible, and they allow a greater range of amino acid changes, so you can have different contact sites. While in between them are these strands, and these strands are like the girders that keep up the antibody so it holds up in space. And this is my impression of an antibody. This is the heavy chain, and it has 3 hypervariable loops, and this is the light chain, and they are juxtaposed. And antigens will bind somewhere at the confluence between them and have between 1 and 4 of these loops actually have contact sites. So it's a tremendous Swiss Army knife of a system, and it's fantastic that the whole thing is built on the same framework. And the point is, as we're going to get ultimately to understanding germinal centers, the process is unbelievably fantastic because if it's not autoreactive and the B cell survives, the genes are there and it gets in the periphery and it actually finds a little slot in the peripheral pool. The point is that then if it encounters T cells, it will actually have this system turned on where a set of enzymes originally designed to repair DNA breaks, which occur all the time, you're exposed to UV radiation or whatever, causes random mutations along this linear strand. And this is a point mutation. And it's really random, but it actually likes to hit these loops more frequently. And sometimes that will make a better antigen binding, and sometimes the whole thing will crash and turn into scrambled eggs, and that's not good for a B cell, so that clone dies. But there's sequential rounds of selection, and this is after immunization with an antigen. In day 7, you start to see accumulations in the daughter cells of one clone, as each of them has slight variations from these point mutations. So you have a repair messed up system for DNA mutations is actually married to clonal selection, and you end up getting better and better and more effective antibodies that are higher affinity, in a nutshell. So let me just point out that there are different B cells in our body. There are those that actually have different specialties, and so not all B cells are the same. There actually are some B cells that are designed, mature B cells, to actually encounter non-protein antigens, so they really don't use marginal center responses. Marginal center responses are for T-cell dependent protein responses. But there are other B cells that actually clear things like polysaccharides on the surface of bacteria. And they actually sit at the edge of the circulation, marginal zone B cells, and they wait for threats that have first got their smallest amounts leaking into the bloodstream, and they're early defenders. But those B cells that are involved in anti-protein responses, like the tetanus toxoid and many of the vaccines that we use, and viruses and the influenza vaccines, are called follicular B cells, and those are the ones that enter germinal center responses, as we'll get to. So this is the maturation scheme. In the bone marrow, you have different precursors, and they actually undergo sequential ranges of selection. This is actually the one that results in either going to marginal zone B cells, marginal zone B cells for antibody responses to bacteria, or they can differentiate, get a different signal, a signal 3, as I was saying before, and they will actually be the kind of B cells that are looking for a little help, a little love in the world that will help them mature and become more antigen specific, and there actually will rapidly turn into, if they get the right signals, differentiate to plasma cells that are the antibody producing factories of the body. I started by accident talking about these mature B cells, which are most of them are made before you're even born. This is a very different kind of a B cell, a B1 cell. It makes most of the IgM we're born with. I actually had a somewhat aggravating 3-year argument with somebody in my division who did not believe that we were born with antibodies in our circulation. And to refine that, we get IgG from mom that goes through the placenta. There's a specific transfer system, but we make our own IgM antibodies. They're present in the circulation. They actually are there and constitutively produced by B1 cells. my colleague said that there cannot be IgM in the circulation when you're born, and I said, why is that? And she said, because I've sent off that test so many times, IgM on newborns, because she studies neonatal lupus, and it always comes back negative. And I said, but you don't understand. They're not saying it's negative, it's just not abnormal. So that took about a year to figure out. So everybody gets some IgM. If your level is too high and the test comes back positive, that means, and you have other problems, it means you're probably congenitally infected with toxoplasmosis, rubella, cytomegalovirus, or herpes. So she keeps getting negative tests, but nonetheless, and none the more, the IgM antibodies that arise in a sterile environment are there and ready to protect us. Okay. All right, so there's a key concept which I'm really getting to, which is, and you have to replay that phrase about Carl Sagan, we have many, many different B cells and T cells, and they have many, many different binding specificities. And in our bodies, I'll just say that the estimate is that we have about 10 to the 11th lymphocytes of each kind. So that's more than Carl Sagan could conceptualize. And the point is, for an effective immune response, the antigen-specific B cell has to find the T cell that's actually recognizing a little peptide, a little protein part of the same antigen. And if you figure that your circulation only goes at a certain rate, and each was looking for the perfect partner, and they didn't have the advantage of internet dating, how do they find each other? Because otherwise, we would never get an immune response. And that just goes to show you why sharks really aren't very good at defending from influenza virus. So the frequencies are so low, how do they find each other? So this is more or less what I just said, okay? And if you think about how much blood pumps through your heart, through the rest of your body, through all of your organ systems, it might take 100 years for you to actually have an immune response to even the first antigen that you're exposed to, so that would not work. But it's really, really important because here on the left, we have an antigen-specific helper T cell, and at the right, we're assuming that we have a complementary antigen-specific B cell, and we assume that they're already committed to their antigen receptors. And you should just know as background that this B cell, if it encounters its antigen, will take it all up inside, chop it into little pieces, and many of them will be presented in the context of MHC class II to the T cell. And so that T cell, because it's the right partner, recognizes the cognate antigen. So it's fantastic if they can find each other, because they each have different kinds of signals that will reinforce the other to cause those, otherwise they would just have signal 1 and they would die. They provide signal 2 to stimulate the other, and as I like to say, each of these partners provides exactly what the other needs. They are the perfect couple. So this is too small, but you have it you can access this in the slide set. This just goes to show you the diversity of major sets of T cells that we have. They've had different signal 3s, and the concept of Th1 cells that bias cytokine production when they're stimulated, to Th17. There's even Th9 cells, Th22 cells. Boy, it gets so complicated. What is with that? 1 and 2 was enough. And then we have suppressor cells that are T regulatory cells. And then we have something that we're beginning to appreciate more and more that we're calling T follicular helper cells. And those are T cells that do their work in the germinal center. So the properties of T follicular helper cells, they are absolutely required for T cell dependent germinal center responses. We used to just think about helper 1s and 2s. It becomes more complicated. These are CD4 positive T cells. They live in a particular place in the follicle, and they have a particular chemokine receptor. So how do they find each other? The answer is coordinated expression of chemokine receptors, which basically, once they're expressed on the cell sort of wiggles around semi-randomly, but will go ultimately on towards an increasing gradient of a chemokine, and that's how they find their way in the body. They can swim through the bloodstream, swim through the tissues, and ultimately they can actually find their way through the roadmap of the body, and they'll be following a scent that will bring them to the germinal center. So these follicular helper cells, They are much more efficient at inducing a B cell to actually differentiate to make antibodies, and they also turn on this point mutation process that's required for affinity maturation clonal selection, and can also induce class switch from one isotype to another, and each has a specialized function. They do this in part by affecting the B cell through different types of surface molecules like ICOS. They actually are regulated in turn through receptors like this programmed cell protein cell death 1 receptor, which actually tells that T cell to be excited or not, and it relates to whether or not it will aid immune responses or potentially ignore autoimmune B cells. And it does this actually through the capacity to induce different transcriptional factors and cytokines that it produces, as we'll see. So it's all about location, location, location. And at the end of many of our blood vessels, we'll have a junction that actually allows accumulation of lymphatic vessels. So lymphocytes draining from the tissue will go here, and also lymphocytes migrating through the blood vessels will go through in this prototypic lymph node. And it's all organized so that there are places that are T cell rich and B cell rich, and there's actually special doors through the blood vessels called high endothelial venules, and the B cells and T cells find their way to coordinate their efforts. All right, so germinal center reaction provides benefits. It is a specialized anatomic site for coordinated spatiotemporal interactions, you remember that, of antigen-specific B cells and T cells. So they have to find each other in time and space. And antigen-specific T cells are required to be able to provide the necessary second signals in the B cell for hypermutation through a specific enzyme, the adenine imidase. I'm blocking what AID is. Oh, well. Survival, BAFF, B cell activating factor, which we've heard about before, and for differentiation and class switch recombination. And so these do these functions in part through concerted interactions that also involve other cells, and there's a very specialized cell called a follicular dendritic cell, and that's different than a dendritic cell, and it's different than a plasmacytoid dendritic cell. I think dendrite just means branch, so they're just morphologically described, but it's completely distinct. It has very different origins. And so this is actually what a follicular dendritic cell looks like, and it has long dendritic processes. They're even more wiggly-waggly than that. They actually have ichthyosomes, which are wrapped around little pieces of antigen, soluble antigen that goes through the circulation, and they'll stick on there, in part because they have complement receptors and FC receptors for immune complexes. So with adhesion molecules as well, they actually are a place where a B cell can encounter antigen in the tissues and get the right kind of localization efforts, and it can then at this site interact with a T cell that has the right antigen specificity, and we talked about the MHC-TCR interaction, and the second signals through a variety of molecules. CD40 is illustrated here, ICOS we talked about as well. And ultimately, when this is triggered appropriately, the B cell will proliferate and make antibodies. This is a simplified version. Okay, so this is a little bit more complex, but this is the same structure. These are the follicular dendritic cells, and this is divided into two major portions of this arena. One is called the dark zone, and one is called the light zone. It's really very simple names because they're staining for nuclei. So if it's darker, there are more nuclei, there are more cells. Here, B cells proliferate. They get the T cell help signal that they need. It turns them on to proliferate, and actually, as they sort of get tired of proliferating, they start expressing a very specific chemokine receptor that makes them wander up in this region, and they will actually die if they don't encounter their specific antigen. That's basically put on a platter by the follicular dendritic cells, waiting to give them their additional signal. What happens here is some of these cells have had point mutations that I was showing you, the hypermutations caused by the AID enzyme, randomly placed. And so some of these daughter cells, although they may have been initiated from one cell, and there's supposed to be a founder effect, one cell, they proliferate, and each of these may have slightly different antigen receptors. And one is better at binding compared to the other. And so David and Goliath, perhaps, but there's a contest, and the contest occurs up here. And the weakling can't contact the antigen and cannot get the second signals it needs, so it dies a glorious death, I guess. And these guys get tired of being reinforced and stimulated, the winners, so they basically then have different chemokine receptors that end up expressed on their surface, and they wander back to undergo another round of cycling, of proliferation, antigen receptor mutation, and clonal selection. And here it just goes to show you the T-cell rich area, the B cells, will actually start to express one kind of receptor and goes towards one cytokine that's in the light zone and conversely in the dark zone. And this allows a very efficient process for selecting very effective B cells, and they're much more clever than T cells. This shows the same thing. It shows you also that some of these B cells, they just don't have what it takes, and they die a programmed cell death, and then they're cleared. And so the whole process is very concerted and involves, and so this is actually why mammals rule the earth, I guess, and the dinosaurs didn't make it. Maybe not, but, and something else that's part of the system that was recently described was this B cell activating factor, also called B lymphocyte stimulator. We know this as the target for belimumab, so the only drug actually approved for lupus based on randomized controlled trials. And up until recently, they thought it only affected the survival of early B cells. Now we actually know that T follicular helper cells are the only sources in the germinal center of BAFF. Now, that actually is very highly refined and fine-tuned and expressed at low levels in health so that you can have B cell selection. It turns out that this is a system where you have inflammation and there's all the white cells are going nuts for other reasons, infection or injury. Many different cell types can otherwise inappropriately express high levels of inducible BAFF and Blys. And so, in fact, these can contribute to expansions and survival of B cells in a non-selective way. So it allows you to ramp up defenses, I guess, but it also makes you prone to survival of autoreactive B cells that otherwise should die in that germinal center reaction. And again, there are specific receptors for BAFF. Oh, here it is. It just didn't, it got shifted. And there are three different receptors that actually trigger different signaling pathways that are expressed sequentially on different B cells at different stages of differentiation. The system gets more complicated because there's another factor, APRIL. Together they cause signaling through making heterotrimers, and there's different affinities and different requirements for each of these pairs. But they allow, in health, a very refined selection, but also a number of agents have been developed for targeting this interaction. Again, belimumab has proved to be successful and useful in many cases. Patients have lupus. If you look really carefully, you can see that these B cells at each stage, sequential differentiation, have different levels of each of the three receptors. So here's TACI, and this is BR3, BAFF receptor 3, and BCMA. We argue about which is actually expressed at higher levels on cells that are in the germinal center and plasma cells. Just to reiterate what I was saying before, the first immunoglobulin on the surface of the B cell only comes up in the immature B cell stage. So here we see our antibodies on the surface. And in fact, these launch into the bloodstream. They're called transitional B cells, and they hope to live and get into the periphery and get into that recirculating pool when they become naive mature B cells. But next stage of differentiation requires the right survival, as does germinal center B cells, and to be able to be accepted as part of the recirculating memory pool, memory of a prior exposure, so you can ramp up a response much more quickly. So this is way too many words, but the point is T follicular helper cells make Blys. The other thing that T follicular helper cells will make is something called IL-21. And this actually is a factor that causes the receptor for Blys to go down, and so that actually is the next step where it'll cause differentiation and make the B cells go into plasma cells. So each of these are concerted steps with different signals. This is just a graphic production that my friend Mike Cancro made because I guess he had a lot of time on his hands. So immunization, and this is a mouse, results in a gigantic spleen that if you then have a cross-section, you can see what these follicles look like that have T-cell rich areas, and then they have follicles where B cells are there and they're naive after the immunization. you actually have cells that are accumulating that come in through the lymphatics, and they will ultimately result in the expansion of these germinal centers that are bright red. And this ultimately results in the expansion of antigen-specific B cells that we can see here, kind of. And this process involves somatic hypermutation and point mutations that result in the selection, ultimately, of higher affinity B cells on the follicular dendritic cell. The lower affinity B cells will die, as will those that are autoreactive. Higher affinity B cells, some of them will turn into plasma cells that will, the long-lived ones will actually persist in the bone marrow. This is a bone, that's the marrow. And some of them will become memory B cells that will persist for years and years and years. And they're kind of not too awake, they're just vigilant, but if they get recruited into a new response, then actually they can react much more quickly, proliferate much more quickly. And the classical example is age 10, we get our tetanus toxoid vaccination, but we forget afterward to get boosted. But there are examples of people who only had single immunizations, and decades later will step on a rusty nail, but they will not succumb to lockjaw because they ramp up very readily their B cell response and the antibody response that neutralizes the toxin that otherwise would cause this nerve paralysis syndrome. So memory can be very potent. It is not passed between generations, but once you generate it, it can protect you from having a recurrent infection, sometimes to bugs that are really horrible and threatening to the species, like some influenza vaccine strains. So the influenza strain from 1918 that caused remarkable levels of mortality, as well as post-infection neurologic syndromes, Parkinsonism as well. People from that era that survived would never get infected with that strain again, or rarely ever, but we don't have that memory, so we have to depend on the vaccine manufacturers. All right. So this is just to point out, here's the central compartment. Many, many precursors are generated each day. Many of them will be autoreactive just by chance, and they will be deleted. A very small percentage will actually survive to the next stage of differentiation, and a smaller percentage will actually be adopted into the recirculating pool. The system has checkpoints to remove autoreactivity. And even at the level of the germinal center, you can imagine that some of these point mutations will result in autoreactivity, and those B cells need to have the right second signal or they will be deleted. And if they are successful, only the few and only the strongest survive in the memory pool, at least theoretically. Okay, so we're making progress in better understanding what goes into B cells at each stage of differentiation, and these are transcriptional profiles. And so each of these is a different gene, and they're looking at a transcript of a sorted population from humans, and you can see that at each sequential stage, different sets of genes are expressed, and they're beginning to help us better understand how, in fact, B cell regulation occurs, going from the earliest theories of Paul Ehrlich and then Mel Cohen, who only seems as ancient but is a bit younger. And so I'm going to just close on a comment about plasma cells, and so there's increasing interest in plasma cells. You continually generate with each new response plasma cells, but many are short-lived. But the ones that are long-lived can be very important for protecting you. And there's, we do studies where we actually survey for these kind of things, and I can tell you that 80+% of the people in the room, in your blood, will have very substantial levels of antibodies to tetanus toxoid. And even though you may not have been exposed for years and years and years, that memory persists and it protects you, as well as the B cells that are somehow behind that, ready to actually be expanded if they're ever threatened. And that system is just being sorted out right now. And you can see that here we have a plasma cell. It has an insane level of endoplasmic reticulum. It is spewing out antibodies all the time. It has no surface immunoglobulins, so it really doesn't care what antigens in the environment. It is just a machine for spewing out antibodies. But these B cells, these plasma cells, excuse me, wouldn't live very long unless they had the right local contacts. And it is not through their immunoglobulin receptors. It actually needs survival factors like in the bone marrow, IL-6 and IL-21 and chemokine receptors and a little bit of pinch of this and a little bit of pinch of that. And this will actually cause expression of different transcriptional pathways. And actually, it continues to need to express the receptors on its surface. So how we can promote plasma cell survival is very important. And we should also think about that in autoimmune disease, you may well have a disconnect between the antibodies in your circulation and whether or not you actually have synovitis, let's say. So you could have ACPA production, but your synovitis is gone because you've been effectively treated by your brilliant rheumatologist, but those antibodies are just going round and round and round, and they mean that that shows you that you have pathogenic autoimmunity that's still in your body, and that there may be memory B cells. So that may be a whiff of past pathogenic autoimmunity, and the risk of reactivation goes on and on and on. So other diseases like lupus, we can have a similar scenario, and some of those autoantibodies may be pathogenic in themselves. So the phrase was developed years and years ago. Can we wipe the slate clean, the tabula rasa? And once you've been exposed to the right antigens with the right genetic background, you develop pathogenic autoimmunity. Can we actually ever attain drug-free remission? Well, I think the argument would be it'll take a lot of things, and it'll include how we can wipe out antibody production from long-lived plasma cells. But good memory, good plasma cells versus bad memory and bad plasma cells, how do we get rid of the chaff without throwing out the wheat? I think I said it right that time. Okay, so this is just a comment about BAFF. BAFF in many lupus patients is very significantly elevated, and this may contribute to the flaws of immunologic tolerance. Rheumatoid arthritis patients are generally not that high. Many may still be higher than normals. At the site of disease, very high levels may be produced. This is synovial fluid. And this is different ways that we actually censor bad autoreactivity at each stages. And it may be very complex, and their defects can occur, at many levels. And there are human diseases, I should say, that are associated with aberrant T follicular helper cells. And I have to say, in many cases, you don't know if this is the cause of the disease or the effect of the disease, but some patients with lupus, Sjogren's, RA, juvenile dermatomyositis, even thyroid disease, and myasthenia gravis all appear to have abnormalities and dysregulation of their germinal centers, which are really the cauldron for making better defenses, but if they're aberrant, you can't weed out the pathogenic autoreactivity. This is just to allude to the fact that diseases like rheumatoid arthritis, a large proportion actually have lymphocytic infiltrates at the site of disease, the synovium, and you'll have B cells and T cells that are not supposed to be in your aching knee. And sometimes they actually take on the features, at least, of germinal centers, and people discuss whether or not that's where the flaws in tolerance actually occur. So we may need to wipe those sites out as well. Now we get to our audience participation phase. So which are you going to vote for? Which is the incorrect answer, please? The others are correct. Architectural structures and secondary lymphoid tissues, which are prevalent immune responses of all species after the emergence of amphibians. Is that the incorrect answer? You guys all get to vote with your interaction toys. Okay, this is not a keychain. Sites where there are highly choreographed interactions of antigen-specific, B cells and T cells. Sites that support proliferation of B cells in response to antigen exposure. Or D, a site associated with lymphocyte programmed cell death. E, sites associated with induced class switch recombination and memory B cells. Which is the incorrect answer? Vote. Bum, 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 ba-da-da-da-da-da-da. Okay, that was too quick. The incorrect answer is, that is correct. I am a mammal and I am proud because I got germinal centers. All right, so A is the incorrect answer. Which of the following is not true for the range of carrots and sticks that helper T cells can use to direct, expand, and select antigen-specific B cells. Which do they not use in germinal centers specifically? I don't know that this is that easy. And you're right, I forgot to talk about fast ligand. I realize that. But truthfully, APRIL is not clear that that actually is expressed in the germinal centers, but BAFF is certainly very important. Fast ligand comes in and out of fashion, I have to say. That's more controversial. And the other three clearly are expressed and very important for that coordinated selection. All right. Dysregulation of which of the following is not known to contribute to the development of autoimmune disease? So I told you there was a lot of ways that you may mess up checkpoints and tolerance. Which is not known? A, overexpression of Blys. B, development of ectopic lymphoid tissues and putative ectopic germinal centers. C, functionally defective follicular dendritic cells that do not allow germinal center formation. D, inadequate negative selection of autoreactive B cells. And E, long-lived plasma cells that produce autoantibodies. So which has not been identified yet? Yes, that is probably correct. I don't know everything. You ever wonder whether all diseases have all been identified or not, and so you're never going to have anything named after you? But, all right, so concluding remarks. I'm sorry I've showed too many slides and talked too long. That's my concluding remark. Germinal center responses facilitate highly coordinated B and T cell interactions. If you have a breach of immunologic tolerance to self-antigens, you can actually have worse autoimmune disease because of misdirected germinal center responses contributing to autoimmunity. And understanding B-T cell interactions has highlighted new therapeutic targets. And these are a number of different levels. They've been developed. And I have to say that we're only beginning to truly understand the implications of some of these therapies. And so we've learned a lot about how immune dysregulation can contribute to autoimmune disease, and I appreciate your attention. And laughing at my jokes, I liked a lot.